You can share now, Minaski. Minaski, we can't hear you. Hello, Minaski.
Yeah. Am I audible now?
Yes, go on.
Yeah. Okay. So today, like we're in the name of God. So today I'm going to start the research workshop on CRISPR. So it's all about CRISPR-Cas9 technology.
Can you make it
So
louder,
before
more? Louder?
yeah.
It's
It's all about CRISPR-Cas9 technology. So today, like we are learning about the introduction of what the CRISPR
hello,
technology.
Minaski.
Yeah.
It's not audible.
Now.
No, you are not loud enough. So it's just fainting. I'm just hearing you faintly.
Okay, 1 minute. Okay, now am I audible?
Yeah, go on.
Okay. Okay, so today we are going to see about the introduction of CRISPR-Cas9 technology, and it is one of the promising tool for gene editing. So before moving on to the Cas9 technology, so first we must want to know what is CRISPR. So CRISPR stands for cluster regularly interspaced short palindromic repeats. It is like a simple explanation that it is a technology that can be used to edit genes. So it is like simple and it's a finding a specific bit of DNA inside a cell. So CRISPR-Cas9 technology is a unique technology that can able to make genetics and the medical researchers to edit the part of a genome by removing or adding or altering the section of the DNA sequence. And it is most rapidly evolving technology and it has revolutionized the biological research. And it represents a family of DNA repeats in most in archaeal that is 90% and in bacteria that is 40% of the genome. It can be in prokaryotic. So it can provide us a acquired immunity and resistance to the foreign genetic elements such as virus and virus. And before moving on to the parts of CRISPR, the CRISPR has three systems. The system that it has, like CRISPR 3 stands for Cas3, and that is a type of one system, like Cas9 is type of second system, and Cas10 belong to the third system. Now I'm going to concentrate only on the CRISPR-Cas9 system. So CRISPR-Cas9 system, that is Cas9 system consists of three parts, that is Cas genes. The Cas genes, what is the role of the Cas gene is to unwire the DNA and to cut the DNA at the specific part of the DNA. Okay, so the Cas gene, which consists of 4 to 20 different Cases, and the CRISPR region, that is, you can able to see the CRISPR region here, and which is has a DNA replicates and the DNA repeats, and which also have a CRISPR repeats also. That is a CRISPR array. So that is cluster regularly interspaced short palindromic repeats. So the in between is a spaces which is occupied by the external factor. So in the upcoming slides, we'll be explaining you this model. So before moving on to the model, let's come to know the history of CRISPR. So in 1987, the CRISPR sequence were first discovered in Escherichia coli. They have found that the cluster regularly interspaced short palindromic repeats, where they have found out the palindromic repeats, that is GATTC 5' to 3' and 3' to 5'. And in 2002, they have identified the Cas genes that have been associated with the DNA repeats in the prokaryotics. In 2007, CRISPR provides acquired resistance against viruses in prokaryotics. And in 2012, there is an idea of using CRISPR-Cas9 genome engineering tool, and which was published by Jennifer Doudna and Emmanuel Charpentier. And they got the Nobel Prize, and it was a great breakthrough in the year of 2015. So here are some there are 6 distinct type of bacterial CRISPR system. So now we are going to consider only the 3 main types. In that 3 main types, we are going to consider only one type, that is Cas9. Okay, the upcoming section will be learning about the other systems. So it is very owning like a simplicity. So Cas2 system, which has been adopted by using a genome engineering. So what are the key molecules are present in Cas9 system, that is type 2? It is like Streptococcus pyogenesis. So which the enzyme was took from that is Cas9, which has a guide RNA, that is cRNA plus tracer RNA. Okay, so first we can see the components one by one. So Cas9, that is CRISPR associated. This Cas9 is called as a CRISPR associated protein. The expanded form of Cas is CRISPR associated protein, and it also known as a molecular scissor. So which can able to cut the specific region of the particular DNA. And RNA guided DNA endonucleases enzyme. That is, CRISPR is always a RNA guided DNA endonucleases. It is always guides the RNA. Okay, so it is always associated with the CRISPR adaptive immunity system in Streptococcus pyogenesis. Okay, so the adaptive immunity, as you know before, like for example, like if you take a COVID virus, when we affected with the COVID virus, a copy of virus that can be stored in our body itself, and it will produce the antibody. Next time when the particular virus attacks us against us, it will release an antibody. It will prevent from getting that diseases. So the Cas9 contains 2 nucleus domain. One is called as H and H domain, and which are very complementary to the cRNA guide sequence, and RUBC like domain keep the non-complementary strands. So now I'm going to explain you what is Cas9 protein, what are the H and H domain, how it can be prevented. So can you able to see my screen? Whether am I audible?
Yes, ma'am, we can see your screen and you are audible.
Yeah. Thank you. So as you can look upon to this image, like can you see when a virus particle, when a virus that infects a bacterial cell, okay, there are 3 steps that can involve in it. One is adaptation, another is production of CRISPR RNA, the another is targeting. So the first step is about the virus which infects the bacterial cell and it releases the DNA into the bacteria. So the DNA will get adapted to the CRISPR region. So this black color diameter symbol is called as the CRISPR region. That is CRISPR loci, you can say, CRISPR array, we can say. So like when the DNA is chopped into small pieces, that is occupied in the different loci, different phases. So when the DNA is invading the virus and it is like processed into short segments. So that short segments are inserted into the CRISPR sequence. These are the short segments. And these short segments are called as the spacers. Okay. So this diamond shape is called as a CRISPR sequences. That is CRISPR array sequences. And like this, if any of the virus that always infects and it forms a new spacers. So it is one kind of DNA spacer and this is another kind of, so it stores like the information. So when again the same type of virus comes, it will chop it into small pieces and it will get eliminated. So these are the simple steps in adaptation. So next is the production of CRISPR RNA. So the production of CRISPR RNA, it's like a repeats and the spaces in bacterial DNA, so which can undergo a transcription. So this DNA is present in the spacer. So in that spacer, like it will undergo some process of copying the DNA into RNA, that is ribonucleic acid. So which can able to, unlike the double chain helix structure of the DNA, which can able to result in the formation of RNA that can be in a single chain of RNA. So the RNA chain is cut into small pieces or called as CRISPR RNA. So here there occurs a production of CRISPR RNA. So next we'll move on to the, if we have in our hand, we have CRISPR RNA in our hand. So next what we are going to do is the targeting part. So the targeting part which will tells you about the CRISPR RNA guide. So the bacterial molecular machinery which can able to destroy the viral material. So because we have a CRISPR RNA sequence and which are being copied from the viral DNA. So here are our viral DNA. The DNA is copied into RNA and the RNA forms a single chain and it is chopped into small CRISPR RNA, and this CRISPR RNAs are copied from the viral DNA as I mentioned you before. So it is acquired during the adaptation. So there are exact match to the viral genome. So these CRISPR RNAs are the exact match to the viral genomes. So thus it serves as an excellent guide. When again that virus infects, the sequence, the target sequence, the copy of CRISPR RNA go and bind it, and it will cleave, and the Cas9 will come and it will chop the particular sequence, and it will knock out from causing the disease. Mostly when the bacteria is infected with the virus, it goes with the lytic cell, lytic process. So the lytic process, after that it will burst. But anyway, the bacteria will, what it will do, it will prevent the process of blasting. So what it will do, it will protect itself as much as possible, so it can able to survive and it can able to enhance their immunity. So these are the process, that is adaptation, that is the virus infection, the bacterial cell. Next is adaptation, next is production of CRISPR RNA and the targeting part. These are the basic concept of the CRISPR technology.
So I have a question here. Can we
Yes.
ask now?
Yeah, tell me, sir.
Hello, hello.
So my question is, ma'am, that why virus is DNA is when entering in the bacterial cell, why bacterial cell is not like a chop down the DNA, or like how it's not recognized by the bacterial cell?
Sir, actually the virus has some PAM sequence. See,
Okay.
as we have the viruses has some tags. They have some specific tags actually. The
Okay.
virus has some specific tags. It can able to analyze the proteins easily. Actually,
Okay. Yeah. Okay. I will read more about it. Thank you so much.
yeah.
Hello.
Yeah, man.
Thanks, Manaksha. How are you?
Yeah, I'm good. And you?
Alhamdulillah. I have a question actually. It's a two question, and
Yeah, sure.
one of them, yeah. This mechanism, it's just to confirm if the bacteria has the CRISPR gene. This is the first bit, yeah.
Like I didn't get you. Your voice is not audible that much.
Yeah. Sorry. The CRISPR gene, if it's there, that means the bacteria, if it's infected with the virus, will act in that way you speak about.
Uh, Still, I couldn't able to hear your voice. It's like two.
Okay, sorry about that.
Can you just put your question in chat?
Yes, no problem.
Yeah, I'm on the chat. You can...
Rukaya, you have an echo. You have an echo on the ground. Rukaya, you can type it and send it. Are you sending? Is she sending a message? Minaski, can you? Okay, I think she's sending it right.
First question: this mechanism, if is infected in bacteria, only has a CRISPR gene. Okay. Yeah. So like yeah, if it has some CRISPR RNAs, it can do that. If that particular bacteria has the CRISPR technology, that is the CRISPR sequence, we can do that. This mechanism is not infected in the particular bacteria. You can also work with certain bacteria which have the CRISPR sequence, CRISPR genes. There are many works are going on in different bacteria. Like you have to do some research on it. So in upcoming class, you will be doing that. Yes, it is like an adaptive immunity. Now this technology is going to come for the human disease also. So now they are planning to do some research. The research work is going on for human also, because human also have some adaptive immunity as well as innate immunity. So CRISPR is act as an productive mechanism, productive immunity against viruses. So here, like as Sir asked me the question, so CRISPR, why can't we directly chop off the DNA? So the DNA has been unbinded by using Cas9 protein. Okay, so we are considering only the one strand, and it can be chopped. It can be chopped into small pieces. That small pieces is get into the DNA, and it can be measured as a RNA, sir. Whether do you have any doubts, sir?
No, ma'am.
So you didn't got my point?
Yeah, I got your point. I got your point. I understand.
Okay.
Thank you.
Okay, so Cas9 contains two nucleus domains, HNH domain and RUVC-like domains. So HNH domain will always cleave the stand complementary to the CRRNA guide that the guide we have processed, and RUVC-like domain which will cleave the non-complementary strands. And Cas9 protein induced double stranded break, as I mentioned you, like it will create a double stranded place at a particular place. So like as you can see, I will show you to the next slide. Okay. Okay. So guide RNA. So guide RNA represents the complement, that is CRRNA, that has been copied from the viral DNA. So tracer RNA. So tracer RNA refers to trans activating RNA, which is the hybrid of CRRNA. So that together forms an rRNA. So cRNA, which consists of 20 nucleotide sequence, which are complementary to the target region and in the specific genes. So tracer RNA provides a scaffold for the complementary RNA binding and for the stabilization. So CRISPR immunity, mediated adaptive immunity. So immune system is created by CRISPR in the bacteria, which consists of two phases. One is immune phase, the other is immunization phase. So now we are going to deal with the phases, immune phase. So first, a virus that infect the bacteria. So that that virus is chopped into small pieces, which is a primary response, and it is inoculated. Inoculated in the host DNA. That's a CRISPR region that is inside the spacers that are available. If it's like for example, like a new virus is affecting, means it will again create a new spacer here. Like it will create a new spacer here. It is already an existing a virus copies. It is like a new virus, so it will add the spacers. So DNA fragments, these are the DNA fragments that have been inserted in spacers. That is earlier the viral infection occurred. So that's how, as I told you. So the secondary response, what it happens is it will go for the transcription. It will from the DNA, it will form the RNA. So the pre-cRNA, and it will chop up into small pieces, and it will bind with the Cas9 protein, and along with the Cas9 protein, it will also bind with the tracer RNA, that is cRNA plus tracer RNA. So this together activates. How it cleaves? It cleaves by RNase 3, that is ribonuclease 3. So it cleaves by using the ribonuclease 3, and for activating Cas9, this cRNA and tracer RNA guide the Cas9 to the particular complementary, to the particular specific sites of the DNA. Okay, so it will go and bind, it will cut in the particular specific sequence. So as you can see here, so this is a foreign DNA acquisition. So DNA binds, and these are the Cas genes. So these are the repeats in the spaces, and this called as a CRISPR loci. So we have a transcription process, and we got the DNA from the RNA. This is a pre-cRNA. So the pre-cRNA is processed into CRISPR RNA, where your CRISPR RNA plus tracer RNA has been processed, and it can help to guide, it is act as a RNA guided targeting for the viral element. So transcription and the translation of Cas9 genes. So translation of tracer RNA. So as I told you before, tracer RNA binds to the CRISPR RNA and forms a gRNA, that is guide RNA. So this guide RNA, it is attached to the Cas9, as you can see here. So these are the Cas9. Okay. So it forms a complex, as you can see, cRNA plus tracer RNA and Cas9. It forms a complex substance. So for example, bacterial genome, CRISPR RNA, Cas9, Cas genes, cRNA, and in the locus we have cRNA of the particular thing. If again a virus infects, we have a target sequence. The virus will have a specific BAM like a tag, BAM sequence like a tag. It is called as a tag. We can say it is a tag. So the reference sequence will go and bind to the particular, the target will go and bind to the particular CRISPR loci, and this will able to activate the Cas9, and it will also go and bind, and it will cleave the double strand, it will create a double stranded break, and it will cut the specific DNA. So this is the major part of the CRISPR-Cas system. So complex. So cRNA plus tracer RNA plus Cas9 goes through a target DNA. So when again the virus infect the DNA particle will have some BAM sequence, which will help to match the sequence with cRNA. Okay, that is our guide RNA. So it will go and find the particular space and it will chop out. So as BAM is called as a protospacer adjacent motif, it is a short conserved sequence. For example, NGG, N is if in your last, if you have a sequence which have a, instead of N, it has A, T, C, G. Okay. So you can use it. If N, instead of N, you can see A or T or C or G, it can called as a protospacer at the end of the sequence. So maximum the sequence nucleotide will be, the DNA nucleotide will be like 20 sequence, 20 nucleotide you can see. At the end of the sequence, it can be like 16 into 4 like that. So double stranded DNA is cleaved by a Cas9. As I told you, the Cas9 will unwind with the helices and it will cut using Cas9. This Cas9 is activated by both cRNA and tracer RNA, which will guide and go and cut at the specific site. So DNA is repaired by the cell. Okay. So here are the genomic DNA. Here are your PAM sequence. Okay. Here is your cRNA, and here is your tracer RNA. Okay. The backside blue color is your Cas9 nuclease. So like when the DNA enters into the system, it will unwind the double stranded DNA breaks, and at the end you can find the PAM sequence. Okay, so it will create a double stranded break. It will create an unwind the DNA, and it will create a double stranded break and go and attach with a complementary RNA. Okay, and it will chop out the particular specific sequence. Okay. So here you can see the clearly. So here is your CRISPR RNA. Okay, here is your tracer RNA, trans activating RNA, and here is a target genome loci. Okay, and here is a PAM folder. So the DNA is when the DNA enters, it unbinds. It will go and bind with a specific complementary RNA. It will chop out with the help of Cas9 and activating the tracer RNA and complementary RNA. It will chop out and it will forms a forms a clear image. So till here, I stopped the session. So do you have any doubt?
Any question?
So if I take the next session, it will be totally it will be confusable. So we have non-homologous. We have a type of like deletion, insertion types. So we can see it by the next class. So I just don't want to put every concept in one session. So it will be totally confused. So I just want to give you a simple gist. Okay. So CRISPR-Cas9 is like a promising tool for gene editing. So CRISPR technology is a unique technology which can able to help you to remove, add or alter the DNA sequence. So it provides acquired immunity to the foreign resistance. Okay, so it is present in the bacteria that is prokaryotic and archaeal 90%. So in CRISPR-Cas system, there are Cas genes. Cas gene refers to H and H domain and UVC domain, and we have CRISPR RNA plus trans activating RNA, and we have repeats and spacers. So history of CRISPR. So the first CRISPR technology was identified in Escherichia coli, and you can also find the Cas genes were identified in the prokaryotic, that is the DNA repeats. The CRISPR provides the acquired resistance against virus in prokaryote. So Jennifer Doudna and Emmanuel Charpentier are the one who find the genome engineering CRISPR-Cas9 as a genome engineering tool. So there are three main types of CRISPR system: type 1, type 2, type 3. So type 2 type 2 only we are going to consider, okay, in this research workshop. So type 2 is Cas9. In day-to-day research, they are doing with the Cas9. Most research works are doing with the Cas9. So if you want to do your own research, so it should be, your work should be based on your title. You have to design your project. You have to design your CRISPR project based on your title, what you're giving. So there are 3 additional types, that is 4 to 6. So we are going to fully focus on type 2. So in the type 2, we have Cas9 and we have guide RNA, and we have PAM sequence, we have the targeted DNA. Okay, so these are the things you have to keep it in your mind and focus it. So Cas9 is associated. Cas9 is otherwise called as a CRISPR-associated protein, and it's also molecular sister. And the Cas9, which is mediated by a CRISPR RNA, that is RNA-guided DNA endonuclease enzyme. So so here, like I'm only introducing the Streptococcus pyrogenesis. Okay, so in this bacteria we have CRISPR adaptive. We are doing with the CRISPR adaptive immunity system. Okay, so as I mentioned you, there are 2 types of domains. The HN domain will always cleave with the complementary strands, and it will not cleaves with the non-complementary. Non-complementary strands, it only deals with the RUVC like domains. Okay. So guide RNA, as I told you, guide RNA which have like CRRNA plus tracer RNA, which have 20 nucleoside sequence, which is a complementary to the particular the specific DNA. So immune phase, as I told you, these are some basics. So do you have any doubt till here?
There's a question. Check the chat. There's a
Okay.
question.
No from my side.
Does the CRISPR found normally in the bacterial cells, or it is acquired, or it is inserted? It is not inserted, it is acquired. It's not, the CRISPR can be normally found in bacteria. Like there are many bacteria that are available. Okay, we can also search, like we'll see in the upcoming sessions. Okay, In the next session, you will be learning about the other bacterial cells. What are the bacterial cells that involved with the CRISPR system? Okay, first they have found out in Escherichia coli. Same thing they are doing it with the Streptococcus pyogenesis, which we have Cas9. So they're for different bacteria will associate it with the different Cas proteins. Okay. So do you have any other doubt? Yeah, sure. We'll be sending you the recordings, ma'am. So do you have any questions or related to CRISPR? So what about Roger, sir? So do you have any doubt?
No, ma'am.
Okay, sir.
Hello? Hello, can you hear me?
Yeah, sir. Yeah, sir. You are audible.
All right. So yeah, we are going to work on the we are going to get back. So we'll send the schedule because the next class will come up next week. So this today we're just starting today to introduce for introduction. So from next week, the main part is going to commence, and we're going to send the schedule to the WhatsApp group. Okay, on how it's going to run. So if there's no more question, we're going to call it a day. So see you guys next week, and then we'll send the schedule. Require ask the question. We'll send the schedule to you, or we'll send it to the group, probably as a document or as a message, a WhatsApp message. So we'll send it there so that you can know the schedule from time to time. Meanwhile, please try to make a personal research. Try to understand the CRISPR on your own. Make a wide research on it. Make research when it comes to the bacteria. When it comes to acquiring the the CRISPR. So just make a general steps so that you have a full basic and background understanding before the practicals begin. Okay. We cannot just start the practical until you have a basic background of what you're dealing with, right? So you must know what CRISPR is. You must have the basic understanding to be able to know how to apply it when it comes to the practical applications. So thank you so much. See you guys next week.